a wonderful and educational morning, I'd like to introduce Dr. John Kirkwood.
Several members of the team who are behind or in addition to those that Val mentioned already. Richard Dickinson, whom you will all recognize as our photographer in the clinic, is actually running all of the video and other things that we had to orchestrate before. We have both our clinic nurses and our physician assistants and our research nurses and our data managers in the big table that's sort of in the middle on the side. I think you'll all recognize them. And they, who really are a critical part of the caregiving mission that we have, are here and will, like the rest of us, stay to answer any questions that we don't address in general terms in the beginning. And I think on the other side, we have only one of us from surgery and one of us from genetics and epidemiology and one of us from medicine. But I think you all know that there are 25, 26 professors here at the Cancer Institute whose whole mission focuses on melanoma, and that this is a very complex undertaking that we're in the midst of. 24 years here, but many more when we really look at the long haul to try to make a difference in this disease. And I think the mission that we have, one and all here, is to cure melanoma, to make a difference in its morbidity, its mortality. And I think that's a place where you all play a huge role, because the mission that we have, the three-corner hat that we have on the faculty, is partly patient care, and I think you all know that part of it for us. Part of it is research, and we're going to each, in our parcels of the talks this morning, tell you what's new in the research, what we see coming down the pike, try to explain some of the words, the gibberish that we may sometimes present in acronyms to you, and what they mean, how they fit together. But in the end, what we are all here to do in the university is to teach. And many of you think that we are here to teach the other faculty from other places in our travels and lectures. Many of you think it's to teach the fellows and the residents, the students that troop through the clinics. But in fact, the highest mission, I think, that we each, who will talk to you today, have is to teach you as our patients, as our patients' families, as our patients' caregivers, because you will have a far bigger impact on educating the rest of the society about this disease. So with really nothing more ado, I'd like to introduce the first of our three faculty today who will talk. Sancy Leachman is someone I've known for a very long time. There are not many people who can present the genetics, the epidemiology, the molecular biology that's really the revolution in melanoma lucidly, so that even I can understand it, but Sancy can. And I will really turn it to her now and thank her for flying in from Huntsman Cancer Institute in Salt Lake, whence she came late last night.
Yes.
Welcome, Sancy.
Thank you. Well, I'm really happy to be here. I actually had to drive from Washington, D.C. last night, and it's a beautiful country here. I'd never had the opportunity, and it's just beautiful. I was very impressed. So I'm not sure, do you have a pointer? So just kind of use the, okay, I can do that. All right, so the other reason I'm really excited to be here, I'm going to talk to you about the role of gene mutations in new therapy, but what makes that really exciting is that right now, as we speak, we are truly in the midst of a genetic revolution in melanoma. And that brings, it's probably the most exciting time we have ever had in terms of the potential for melanoma to be treated by using this information. And it also offers a huge amount of hope for patients that there may be something coming on the horizon that really has the potential to impact the disease. So I think that this is I hope you'll agree with me at the end that this is a very exciting time in melanoma. I thought I'd start just by telling you ahead of time what I'm going to tell you so you can follow along. All right, so now you see I didn't realize that you guys weren't seeing my slide there. I think this is a really cool piece. This is a little artwork on the genetic revolution. So anyway, that was supposed to go with my little spiel about how exciting of a time we're having. Okay, so the major points. So the major points of this talk, so that you'll know what I'm going to talk about and you can follow along, is first of all, I'm just going to explain a little bit cancer, and many of you are going to know that there's a huge range of understanding in this room. So please, don't hesitate to ask me questions, and don't hesitate to zone out if this is too simplified for you as well. But cancer, at its fundamental roots, is a failure of growth control. And growth control is lost when proteins called oncogenes and or tumor suppressors malfunction. They aren't working properly. And I'll talk a little bit about how that works. Then it turns out that this malfunctioning is fundamentally caused by mutations or changes in DNA in the cells. When the DNA gets damaged, these little mutations happen, and they happen in those proteins, those oncogenes and tumor suppressors. And I'm going to give you 3 examples of 3 important genes, a tumor suppressor, P16, and 2 oncogenes, c-KIT and BRAF. And then the exciting part comes when I talk a little bit about drugs that have been identified that can actually go after those cells that have mutations in them, in the 2 oncogenes. So, as I said, cancer is a failure in growth control. So I'm not sure what kind of cancer this is actually supposed to be, but you can kind of see that the point is that it's growing out of control. It's a big mass of tumor cells that are extending out. And the process of growth control fails. The cells keep dividing. They don't know when to stop dividing. Normal cells know when their division is up, but these tumor cells don't know. Then they begin to develop the capacity to continue to grow and invade the local tissue. After they invade, they develop the capacity to grow their own blood supply and then to spread through the lymphatics and through the blood vessels to other parts of the body. And they won't die. Nothing can kill them. They're immortal. And then, because they're all over little parts of the body, everywhere, and they keep growing and they keep dividing in this cycle, they basically take over the organs. And that's the way that cancer ends up causing problems. So how does that work? Well, it's basically a failure of these 2 proteins I was telling you about, the tumor suppressors right here and the oncogenes. So what do tumor suppressors do? They do exactly what they say. They suppress tumors. The normal function of a tumor suppressor is to stop tumors from growing, to stop the cell from actively dividing, as you see in this section. This is basically representative of the cell cycle where the cells keep dividing and dividing and dividing. And if you have a tumor suppressor, normally it keeps it in check. It's a great check and balance. And you can think about it like the brake on your car. It's providing that halting when you need to stop. The oncogenes, on the other hand, that's the other side of the equation. Oncogenes are normally not very active at all. And they're kind of like the gas, right? They're not as active. But if they get accelerated, if they get activated, then what happens is the same problem. Instead of not being able to stop because there's no brake, in this case, they can't stop because the oncogenes are revved up, like pushing on the gas pedal and it getting stuck. So that's the main way that your normal cells are controlled, and if they go out of control, it's because of a failure in those 2 types of proteins. But how does that happen? And to understand how the proteins, all of a sudden they're working fine, and then in a cancer, when it develops, why is it no longer working fine? You have to have a little molecular biology. So this is your little molecular biology 101 talk here. So it turns out that the way that proteins are made, oncogenes and tumor suppressors, is it starts with DNA. The DNA in every living cell is the coding. It's the sequence, the code to life, basically, and it codes for these proteins through an intermediate called RNA. And that ultimately leads to the production of protein. And so here's just a representative, this is the human body. Every tissue, this could be the skin, right, has cells. And this cell could be a melanocyte, right? And melanocytes have a nucleus, and inside the nucleus are chromosomes. You can see those little chromosomes. And if you expand the chromosome out, it's made of DNA. And DNA has these base pairs, the A, T, C, G. And if you look at areas of the chromosome, areas of the DNA that range in size from fairly small stretches up to hundreds of thousands of stretches, you can create genes, and that's where a gene is. And what happens is, anytime you divide, anytime a cell divides, it has to copy that DNA. And every time that it copies the DNA, there's a potential that a mistake is made. And given the millions and millions of base pairs of DNA that you have to go through, there are mistakes that are made. And when a mistake gets made, it's a mutation. And mutations can sometimes be fixed. We have a repair mechanism in the cell, but if that fails too, and you happen to get a mutation in a really important oncogene or a tumor suppressor, then that oncogene or tumor suppressor fails to do its job, and you start down that path of developing cancer. So I pulled this off of a website I thought was really nice, and it's talking about breast cancer, but it's exactly the same for melanoma. So I thought it was very relevant. And 5 to 10% of melanomas are inherited. In other words, you've got some gene that's making you that it has a mutation in it that you can pass on from generation to generation. And that's what these pedigrees are about. You have the inherited mutations, but you also have acquired mutations, and that's what most people have. The 90 to 95% of people have a melanoma that's acquired. And as you can see here, what I liked about this is it shows all these different ways that it's possible to get one of those mutations. But the fundamental thing is that the DNA has changed. So it can be chemically induced from air, water, food, or the environment. Some tumors are caused by viruses or bacteria. Now, as far as we know, that's not true for melanoma, but it is for some cancers. Radiation, including right here on the top of the list, solar radiation, can cause those mutations to happen. And a failure in DNA synthesis, the process of dividing and copying properly, or the process of repairing any mistakes that happen. All of those things can lead to a mutation in the DNA that causes you problems in the tumor suppressor or oncogenes. So, what are the 3 examples I was going to give you? So I told you I was going to tell you about 1 tumor suppressor and 2 oncogenes. The first one is a tumor suppressor. And remember, tumor suppressors, you're supposed to have plenty of capacity for a tumor suppressor in the cell to keep the tumor suppressed. And if you get a mutation, it means that it's not making enough of the activity of that tumor suppressor. And so one of the major tumor suppressors in melanoma is called cyclin-dependent kinase inhibitor 2A, or the easy name is P16. The oncogene, remember, has too much activity because of the mutation, and the 2 that I'll talk about are c-KIT and BRAF. So this is believe it or not, this is actually a simplified scheme of somatic melanoma genetic alterations. And so what we're going to talk about, as I said, we're going to talk primarily about the CDKN2A. You can see that I've put in red all of the ones that are the stoppers, the tumor suppressors. And then in green are the oncogenes. You have the c-KIT we'll talk about, and the BRAF. And what I'm really trying to get you to understand from this complex slide is not necessarily all these relationships, but to know that there is a relationship. There are pathways that these molecules are fundamentally impacting when they fail. But the main thing that happens with all of them is that you get this unregulated cell growth. So when the P16 tumor suppressor has a damaging mutation, in the case at the germline, so when that happens in an egg or a sperm cell, where you've got that mutation in an egg or a sperm cell, and it can be passed on from generation to generation, you get a condition that we refer to as hereditary type of melanoma. Fortunately, only about 0.2 to 2% of the melanoma population has this kind of melanoma. But when you get that kind of melanoma, oftentimes you see these funny-looking moles, these atypical moles. And if you're unlucky enough to inherit one of those mutations in that gene, in the P16 gene, your chances in your lifetime of getting a melanoma is about 76%. So these people are at extremely high risk for getting a melanoma. Now, the acquired mutations in P16, what I mean by that is when you're out in the sun and the sun comes down and damages your DNA and causes a mutation in P16 that ultimately develops into melanoma, that about 25 to 50% of melanomas have an acquired mutation in this tumor suppressor P16. Now, right now, the sad thing for me, this is the gene I study. I study hereditary melanoma. So this is pretty sad because right now there's no particular targeted therapy for P16. There's some targeted therapies that affect the protein that it regulates, but they haven't been very useful in melanoma yet. So this one is not, we're not going to dwell too long on this one. The one we're going to talk about next is c-KIT, and I'm going to have some questions for you, so get ready, pay attention. You're going to have to answer some things here. All right. When the c-KIT oncogene, okay, now remember this is an oncogene, and usually you talk about it getting activated. But the way we actually found out what this thing does is we found out from inactivating mutations. So I want you to take a look. This is a buck that has an inactivating P16 mutation, and this is a baby that has inactivating. And I think you can see along the midline of the face, there's a little white spot here, and there's a white forelock, what we call a white forelock here. Does anybody know what the name of this condition is? Anybody have any friends or family members that ever have had anything like this? It's called piebaldism. Okay, now, if you were to guess, and I want anybody that has any ideas to speak up, if you were to guess, what is the problem when you don't have a good c-KIT? What do you think the c-KIT is doing in these cases? Any ideas? So what if I were to tell you that normally in development, the way that melanocytes get through the skin is they migrate from the back to the front? Any ideas? No? So it's okay because, a lot of scientists took a lot of years to figure this out too. But what it does is it controls the growth and survival of these melanocytes. And so the melanocytes that don't have c-KIT don't survive as well, and they don't make it around to the front, to the final place. And so you end up with white stripes where there are no melanocytes right down the middle. Now, if that's what happens when you don't have enough c-KIT, what do you think happens when you have too much c-KIT? Well, it really promotes extra growth and extra survival, and it's harder to kill those melanocytes, and that's why they end up turning into tumors. And if you get an increased number of these c-KITs or an oncogenic mutation, then you get a special type of melanoma. It's primarily these acral mucosal, although now it looks like maybe the ones with sun damage can happen too. But you get these mutations, and that activates it, and it makes it revved up, and then you get overgrowth and less control. And what's interesting is that now there's been a drug called imatinib that some of you guys may have heard of. You're fortunate. I don't know if everybody here, I don't have to say this for the majority of the people in this room, but you really are at one of the meccas for melanoma therapy here. Because of Dr. Kirkwood's group, you just have some incredible opportunities here that most people in the country don't have in terms of these novel and advanced therapeutics. And one of these things, and I think he's going to talk about, is imatinib, is one of them, that now we understand what it's affecting, is it's affecting c-KIT when it's mutated, when it's overly active. So now, what about this other oncogene, BRAF, all right? So BRAF, the important thing about BRAF is about 60% of melanomas are mutated, have a mutated BRAF. And the mutation is this one specific site. And what's happened in the recently is that there's a special set of molecules that have been designed to target this molecule, and in particular, that one mutation. So what is all the excitement about this? Well, think about it. If you have an activating c-KIT mutation that normally leads to the production of this type of melanoma, if you treat with an agent that specifically stops the activity of that molecule, you have a great chance to treat this kind of a cancer. And similarly, with an activating BRAF mutation that has this V600E mutation, which affects about 60% of melanomas, if you have a drug, and this drug, some of you in this room may have heard about this drug, the Plexicon or now Roche BRAF specific V600E mutation drug. Same thing. You can basically target those cells that have this mutation with that drug specifically. And so that's why I said in the very beginning, there's a genetic revolution in melanoma. Right now, what we're beginning to understand is enough about how these molecules work, where we can design drugs that will be able to specifically target those problems and will be able to hopefully treat people in a way that's very specialized for the tumor that they have. So I don't want to leave you with the idea that this is all now done, that we have all the answers, because I think that would be misleading you, and it wouldn't be fair to how difficult this process is. We don't understand why when you treat with some of these drugs, they don't work. Why when you treat with some of these drugs, does it work for a little while, but then the tumor comes back? How do you prevent these recurrences from happening? And we don't understand all of the side effects of these drugs. Why do some of the drugs that are designed to knock out cancer actually end up producing some cancer? And so how do you prevent those when that happens? And finally, I think one of the most challenging things that's coming up is, are combinations of targeted agents and immunotherapy going to be helpful? So if one drug doesn't work by itself, can you go back and hit it with a second one that hits the pathway that will also then ultimately kill more of the percentage of the tumor and give you a better outcome? So let's go back to the major points, and then I'd like to answer any questions that anybody might have. Cancer is a failure of growth control, right? Growth control is lost when oncogenes or tumor suppressors malfunction. The malfunctioning is caused by mutations or changes in the DNA that encodes for those proteins. I gave you 3 examples: a tumor suppressor called P16 that is responsible for hereditary melanoma, and 2 oncogenes, c-KIT and BRAF, that have recently had drugs that were developed. And these drugs that are developed to go after those oncogenes affect change because they're affecting the mutated form of those oncogenes. So, thank you. I hope that was clear.